Good morning, friends. I am Dr. Praveen Bankar. Today, I am going to discuss about PCR RFLP technique. This presentation is for the purpose of submission of MOOC assignment. So, friends, as you are well aware about the COVID situation crisis which just happened, and in that situation, we heard a lot about the RT-PCR, PCR technique. So what exactly is this PCR? I'm going to give a brief idea about this PCR. So this PCR technique, as I have given here, PCR RFLP technique, we are going to discuss about this technique. This PCR RFLP is capable of identifying particular repeated DNA sequence from minute amount of the sample material by a specific amplification of the sequence. Based on the enzymatic amplification of this particular DNA fragment, which is flanked by two oligonucleotide primer, which hybridizes the opposite strands of the target sequence. As far as the advantages are concerned, these are very highly sensitive and fast techniques. It requires much less amount of DNA, and it is a totally automatic kind of technique, which required less labor intensively. Basically, there are 3 to 4 steps in each cycle of the polymerase chain reaction. First is a denaturation, where the DNA has been heated more than 91 degrees Celsius so that the DNA strand, it should be separated. Then the separated DNA strand, they are being cooled by annealing. The temperature of annealing generally about 50 degrees Celsius, and where the single-stranded oligonucleotide primer, which are specifically designed to flank the region of DNA of interest. And final stage is as extension or final extension, where the primers of various DNA polymerase, they used to tag at 72 degrees Celsius. So I will summarize this topic here, that one cycle of PCR doubles the number of target DNA molecule, which are newly synthesized strands, and themselves act as a template in next round of amplification. Each cycle essentially doubles the amount of DNA fragment which is produced in previous cycle, and it is very highly sensitive and fast, which require less quantity of sample, which is very much benefit or advantage. So these are the references for the topics. Thank you very much.